Look closely at a typical neighborhood sidewalk after a rainstorm. That ordinary puddle is teeming with microscopic threats waiting for a host. In common areas like dog parks or sidewalks, highly contagious pathogens are everywhere. Canine parvovirus can survive on bare surfaces for months, while canine coronavirus spreads easily through traces of infected feces. Then there are environmental bacteria like leptospira, which thrive in damp soil and stagnant water. As you can see in this veterinary text, these bacteria are actively spread by urban wildlife, specifically rodents like the Norway rat, which contaminate the environment through their urine. The everyday outdoor world is inherently hostile to an unprotected dog. Surviving it requires proactive biological defense. When puppies are born, they aren't entirely defenseless. They receive a temporary shield of maternal antibodies directly through their mother's milk. But this protection doesn't last. This graph plots a puppy's age against its natural immunity levels. Notice how the line for maternal antibodies drastically drops off right around the 6 to 8 week mark, creating a steep vulnerability window. If a puppy encounters a systemic or respiratory virus during this window, the consequences are severe. Diseases like parvovirus cause aggressive vomiting and bloody diarrhea, requiring expensive veterinary treatment, and are frequently fatal within days. To bridge this gap and keep the animal alive, we have to artificially train the puppy's immune system. We do this using a multi-disease vaccine, introducing the body to these pathogens before a real infection strikes. The moment that fluid enters the tissue, an incredibly dense payload is delivered right into the dog's body. The 9-in-1 vaccine carries 9 distinct threats. The first group contains the heavy-hitting systemic and respiratory viruses: canine distemper, adenovirus types 1 and 2, parainfluenza, and parvovirus. Next, it includes a specific component to defend against canine coronavirus, targeting gastrointestinal illness. Finally, this organizational chart breaks down the last group: 4 distinct environmental bacterial strains of leptospirosis: Canicola, Grippetophosa, Icterohemorrhagiae, and Pomona. All 9 pathogens are clustered here into a single dose. Safely engineering a single vial to hold 9 non-interfering components requires immense pharmaceutical precision. It demands industrial-scale manufacturing and highly controlled environments to ensure the biological agents remain stable. This small vial is a highly complex engineered biological delivery system carrying multiple distinct threat profiles simultaneously. To handle the most dangerous viruses, like parvovirus and distemper, the vaccine utilizes a modified live strategy. Here, you can see the weakened virus bumping into a white blood cell, completely unable to infect it. These viruses have been intentionally stripped of their ability to cause actual disease. However, their physical structure remains intact enough to trigger the body's biological alarms. The vaccine takes a different approach for the leptospira bacteria and the coronavirus. These components are inactivated, relying on completely heat-killed bacterians and dead viral particles. Whether a pathogen is modified live or completely dead, the dog's white blood cells operate the same way. They scan and recognize the unique outer structures of all 9 of these foreign invaders. Once recognized, the immune system responds by manufacturing highly specific antibodies to bind to these structures. More importantly, it builds a permanent armory of memory cells specific to each pathogen. The vaccine runs a highly realistic biological fire drill, safely programming the immune system to identify and fight these threats without ever exposing the dog to an actual illness. Fast forward a few months. The vaccinated dog is fully grown and taking a walk. It stops to drink from a puddle contaminated by rat urine, unknowingly ingesting live leptospirobacteria. Inside a blood vessel, leptospirobacteria attack the vessel walls, causing a threat known as leptospiremia. But thanks to the vaccine, memory cells instantly recognize the pathogen. The immune system immediately floods the blood with targeted antibodies, neutralizing the bacteria before the walls are breached. Clinical data confirms this rapid response works. It effectively prevents both leptospiremia in the blood, and it prevents leptospiruria. This is when the bacteria manage to successfully colonize the kidneys and shut into the urine. Thanks to this immediate recall capability, the deadly pathogen is completely destroyed before the dog ever displays a single symptom. This biological armor naturally fades over time as the immune system's memory wanes. To keep the defense strong, annual booster shots are required. They act as essential refresher courses, ensuring antibody levels remain high to fight off unexpected exposure. When we prevent zoonotic diseases like leptospirosis and rabies in a dog, we halt the spread of pathogens that would otherwise be shed back into the local environment. Maintaining the 9-in-1 vaccination schedule creates a vital biological shield. It protects the dog, safeguards the family, and defends the community from outbreaks.